Thanks for that kind introduction, and thanks to ONT for having me here. So we're going to do things a little bit differently now. I am coming to you all from the year 2043. So we have just fast-forwarded 20 years. So join with me in 2043. So we are on Mars. So we first landed on Mars in 2040. SpaceX Starship, which is how we got back to the moon, and I'll get to that. But we are here. So we first landed on 2040, and that was just a quick boots on the Martian surface kind of mission, but now we're back in a sustained way. And what I mean by that is when our crew got back, they found waiting for them prefabricated habitats with advanced environmental control and life support systems, the power and communication infrastructure they needed, a shelf-stable food system, which was no joke, that took a lot of development, as well as what we're all most excited to talk about, a suite of scientific hardware. So while great things were happening on the Martian surface, what I'm really excited to talk to you about is what is happening inside the habitat. So we have inside our Martian habitat the MinION Mk1X and the Power Blade, which we have automated for sample prep. And so really what this has allowed us to do is things like monitoring of crew health. And I'm going to speak about how we got to that. I'm going to tell you about a lot of firsts. And then another thing that we really had to define before we got to Mars was efforts involving planetary protection. So both forward contamination, what we could be potentially putting on Mars, and then backwards contamination, what we didn't want to bring back to Earth or have our crew get infected with. Monitoring plants. So we learned from early days on ISS that growing plants was both provided a nutritional benefit to our crew, but also a massive psychological benefit. And then I mentioned those environmental control and life support systems. We're recycling everything. Urine is recycled for water, the atmosphere is turned over, so great places for microbial contamination. So this, we will talk more about, but this speaks to the first true application of nanopore sequencing in space, which was microbial surveillance. And then the thing that we all are so excited about, are we alone? And so we've sent rovers, we've brought samples back, but we have not been able to definitively answer the question, is there life beyond Earth, at least on Mars? And so with the suite of hardware, now that we have crew there, we're going to be able to answer that. But how did it all start? So that's where we are in 2043, but how did we get there? So what most of you have probably heard about is that in 2016, astronaut Kate Rubins became the first person to ever sequence DNA beyond the bounds of Earth. And this was a sophisticated experiment for the International Space Station, but really pretty simplistic. The samples that Kate was sequencing were prepared on Earth. So she was doing kind of a thaw them out, load the flow cell, and see how nanopore sequencing worked in the microgravity environment of space. But what most people don't know is before Kate had even done that first run on board ISS was that we had a crew of astronauts and scientists located on the ocean floor in a habitat called Aquarius. So this is off the coast of Key Largo, Florida, where they had already demonstrated our swab to sequencer method. So this was the method that my team developed to really, with the end goal in mind of really being able to understand the microbiome better. So as you hear it, swab and go directly to sequencing. So this happened before Kate ever even did that first sequencing on board the ISS. The following year, in 2017, we were able to transition part of that method to the International Space Station. So we were lucky enough to work with astronaut Peggy Whitson this time to implement part of that. So this is the one time where I'm going to take a step back and kind of dig in just a little bit into the data, because this really was the paradigm-shifting moment for us. So what we did was part of the Genes in Space 3 investigation is, I told you with Kate samples, yes, nanopore sequencing works, but what good is the nanopore sequencer if you can't prep your samples in whatever extreme environment you're in? So what we did was we took, I'm sure it's near and dear to many of our hearts in the microbial community, the Biomics mock community standard, and we just diluted it down. And we sent it, and we had Peggy amplify it, do a cleanup, and then prep libraries and sequence. And we were thrilled to see that her sequencing on board the space station looked just like our ground controls when we had done that the identical split sample on Earth. So this gave us that comfort that, okay, the crew can do this. They can do this sample prep. So the next thing we wanted to do, so back in the beginning of station all the way up until the early 2020s, we had been doing microbial monitoring by having the crew actually culture microbes. So they would collect samples from air, surfaces, and water and culture them. And this did a lot to help us inform our microbial risk assessments, but what it didn't allow us to do to know what those microbes were in real time. The samples had to come back to my lab. So what we did was we had Peggy actually open one of those culture plates, and we said, pick any 3 colonies. And she did, and she put them into our method using mini PCR as kind of our heat block and to do traditional PCR and see what we got. And so from those 3 colonies, after we filtered the data, we had 2 IDs, Staphylococcus hominis and Staphylococcus capitis. And if you're not a microbiologist, that means nothing to you, but it was really exciting to us because this makes sense. This is what we always see on the type of bacterial growth media that we provide. And this is coming from humans, and again, was really exciting, but were they accurate? So we got that exact same plate down that Peggy had used, and she had circled the colonies. So we put them through our standard operating procedures for the lab, which was Sanger sequencing, and then biochemical analysis with the VITEC to get those identifications. And we found out that indeed, 2 of those colonies were Staph hominis and 1 was Staph capitis. So again, this was just like the most exciting moment because it really changed that paradigm that if you want to do science in space, you have to bring it back to Earth to do your analysis. So this really led to a significant increase in space-based genomics that really took off rapidly throughout the late 20-teens and early 2020s. We continued that because if you remember, when I was talking about at NEMO, I mentioned a swab to sequencer method. So just by the nature of culturing organisms, right, we're increasing the risk to our crew because we're increasing the number of potentially harmful microbes. So we wanted to remove culture from it completely. So in 2018, we were able to put our entire swab to sequencer culture independent method on board the space station under the best payload. And the best payload really gave us an umbrella to do a lot more. We did so much, and again, I'm just mentioning first. So we did the swab to sequencer method. We showed that the crew could sequence RNA directly. They prepped the RNA. So we set up native RNA. They did all the prep and sequencing. And this was really exciting because we had heard over and over again, yeah, Kate Rubens, of course, she's great. And Peggy, oh, she's great. All of our crew are great, but can they sequence and prep RNA and it not be a big degraded mess at the end? Yes, they can, and we showed that, which opened the door for all kinds of additional assessments that could be done. We also did cDNA sequencing. So just a lot of capabilities that we were showing and developing in the late 20-teens. From there, with all of the success of that work, all of that was research. So NASA calls those research payloads. So just like the research you do in your labs. But after the success of all those, we moved from being a research payload to actually being medical operations hardware, meaning something we use routinely to be checking on the environment or some aspect of crew health or the space station. And so really, this became known as Biomol. And Biomol was really just that exact same swab to sequencer method from BEST put on board and to allow us to do a lot more. And what was really exciting for us was it allowed us to start to see the true microbiome of the International Space Station versus the very kind of biased view we had been seeing from the culture data. And so again, it gave us this umbrella to do a lot more and demonstrate a lot more. One of the things was just more crew, right? So things that we didn't know as when you're trained as a scientist, that you don't know how to write procedures for a crew, how to package things so it's easier for them to use. And NEMO taught us a lot of that, but also more experience on the space station. So a really cool moment with Kate Rubens when she went up on her second mission and she got to now apply what she had done and what she had started back in 2016 and really do the full swab to sequencer method, but as well as just getting so many more crew members' hands on it and building up confidence and being able to do these pretty manual methods to go, again, all the way from sample collection to DNA sequencing. Also under Biomol, we were able to start to bring in more sample types. So we had obviously been doing surfaces with the swab, but now we started doing water, which was great because our water reclamation system on board, we've never really known what's in it because by the time a sample's collected, it sits around for a while. by the time we get it back on the ground, that microbial profile has shifted completely. The other thing, we got to test out at the time, the MinION Mk1C. And so this is one of our crew, Jasmine, with the Mk1C. And before we had the Mk1C on board, which was great because we could downlink much smaller files after doing the base calling, but we had actually shown data analytics capabilities. So when we were still working with the Mk1B, we had the data transferred from the space station computer where it had been collected to Spaceborne Computer 2, which was developed by HPE. And so this was a collaboration with HPE and IBM and their edge computing. We had taken our pipeline and containerized it and put it on there. So the crew were actually able to go from their swab that they had done all the way through to that microbial taxonomy, as well as some other metrics we had them look at. So this was a pretty big defining moment, because before, when we send out our microbial reports from that culture data, you're looking at 6 weeks, 8 weeks since the crew drank the water, the thing was growing on the surface. And now, with this, we were able to show within 24 hours, what were the contaminating microbes. So again, just in a pretty quick time, we were significantly moving the needle forward. So thinking about even larger, going beyond swabbing inside of the ISS, in 2024, we got to be a part of an EVA to swab the outside of ISS. So this was the microorg EVA. And what we were looking for here is that to inform planetary protection. So this would be, right, that forward contamination of Mars. So we intentionally are leaking ISS atmosphere overboard. We know the suits leak, we know the vehicles are leaky. And so what we had the crew do, it's kind of hard to see in the picture. You can see the large swab that we used for this. That's the crew practicing in the neutral buoyancy lab. And then one of the main areas that was swabbed was right as they come out of the airlock, they go through this vestibule, and that's what you see in the bottom picture. And that's a very high touch area. There's parts of it that are shielded. There's also, when we're venting other things on board, there's nutrient source from CO2 and nitrogen venting. So really good places. And the information we got from this was critical to really informing a lot of those planetary protection models that we needed to get us to Mars. In 2026, so we got to launch our viral React payload. So this is kind of where I'm just, again, taking a small step into the crew health realm. I am a microbiologist, so not going to talk to you about all the work that's been done to use this to looking at astronauts tracking of various biomarkers, but one thing my team did was we know our immunology colleagues have validated that the crew reactivate certain viruses, ones that most of us have that are dormant, and they're shed in saliva, which was a very non-invasive substance to work with. And so we developed a method to be able to track that reactivation of these viruses in the saliva. The gold standard for this was a qPCR assay that always happened post-flight. So this was our first chance to actually be able to start to look at this in flight. And our immunology colleagues had already shown that this was a direct indicator of stress. So when the viral loads are going up, you can see overall other indicators of immune stress also going up. So the idea was, and that we tested this in 2023, 2024 at Palmer Station, Antarctica with their winter overcrew, we would see the viral levels going up, and we would give the crew some kind of a relaxation countermeasure, VR being one of the examples. And then we would assess again to see if those levels had come back down. So again, just one example of the things that we've been able to do in kind of a biomarker tracking to be able to make sure our crew are healthy. So taking a look at MinION's time on ISS as a whole, a 14-year run, pretty good. And with that, we validated methods that I've already mentioned for amplicon, direct RNA, full genome, metagenomics, but really bringing in more sample types. So colony swabs, filters, cells in liquid, tissues, all things that we have been able to process on board. And then also demonstrating those data analytics, the biomarker detection, and genome assembly all on board. So really great things that the International Space Station gave us, but obviously we had to get to Mars, so we weren't stopping there. So 2026, back on the moon. So very exciting for those of us who didn't get to live through it the first time, like Gordon was talking about this morning. And so I'm sure all of you remember where you were in 2026 when Starship landed with humans on the moon once again. So this was the Artemis 3 mission, and this was a quick mission. Get there, get reestablished on the moon, test out some new technologies, but we weren't sustained at this point. To get to a sustained lunar surface operations, we use something called Gateway. So Gateway, just think of a smaller ISS that is in orbit around the moon, and Gateway does two things for us. It allows us to have multiple points to be able to access the moon's surface, because instead of that Starship approach, which was like Apollo, where you launch from one side on Earth and you land at one side on the moon, your crew, your lunar lander is already docked at Gateway, and your crew launch in a separate crew vehicle, dock at Gateway, and then they can access different parts of the moon based on where Gateway is in orbit around the moon. The other thing that we did, those pre-positioned habitats that I mentioned earlier, those were all built in this Gateway space, so as well as our Mars Transit vehicle, because much less upmass to be able to assemble those things there versus trying to launch them fully. Gateway was also really important for our microbial monitoring efforts with nanopore sequencing. So from 2027 through 2028, this is when we were really using known as the microbial surface monitor now, because at NASA we change acronyms a lot. And this was just that BEST and that Biomol method now being used to monitor the surfaces and the water method of Gateway, which was really cool because with ISS, we didn't get to watch the microbiome evolve. We did through culture, but it was a pretty limited view, but with Gateway, we really got to watch and see what the impact was of having crew there for a short time early on in Gateway to building up longer term stays. The other thing is this is when we brought on PowerBlade, that automated sample prep that was developed by the National Research Council of Canada, and this is where we started using it operationally in combination with the MinION. And this allowed us to just increase throughput so much because we didn't have the hands-on crew time that we had when they were doing our manual method. We continued lunar surface operations through 2028 through still today. Even though we're on Mars, we are sustained on the moon and we're continuing those operations. And I really can't, just like ISS did so much for us and gave us so much, so did the lunar surface. And so while we had really great things happening, again, just like Mars, on the lunar surface and exciting discoveries to be had, what's really exciting, again, is what's happening in the habitat. But a quick shout out first to all the women that have been involved in having women on the moon for the first time, and just how critical that they have been in all of our efforts and some of the great science they've given us. And so with that, this is a look inside the lunar habitat. So a few things to notice. You can see the P2 is displayed, it's deployed. I don't know if you can see it, it's stowed on the side, but the Mk1D with its tablet and stowage. So I'm the microbiologist, we're still using the Mk1D, but we've been using the P2 for extended, the P2 Solo for extended applications of nanopore sequencing on the moon. And I didn't have time to go into it today, but we have the P2 Solo and Mk1Ds all the way through the Mk1X in orbit around on commercial space stations that are in orbit around Earth that took the place of ISS. So just some things that we got, some science that we got from the lunar surface. So I mentioned this, again, this idea of planetary protection with the back contamination of Earth. And I get asked all the time, how will you know? How will you know if there's something that's potentially, contaminating if we don't know what it is? And so what we started doing later on in our lunar surface days was doing an almost daily microbiome baseline for each crew member and from the environment. And this sounds like a lot, and it was a lot, and it was a lot to work up to, and that's why the automation was so key. But we could track the baseline. And when we would see perturbations in the baseline, this gave us an indicator that, okay, maybe there's something going on. So that is how we're approaching Mars. If we see perturbations to these baselines that we're continuing to collect, this is one of the ways we have as an alert for that there might be something contaminating. We also were able to go back to the original Apollo lunar landing sites where we had left a lot of biomass. I mean, a lot. And by that, I mean loaded diapers. So, which were, may have not been the funnest thing to go back to, but it was great for us to be able to collect that data because we could go back to the source and then track out, okay, how far can we detect, the DNA signatures of these things as compared to the source. And then that data was critical to being fed into the folks that do the the modeling of the weather and patterns and things on Mars. So complicated problem because the moon doesn't have weather patterns, but the question of dispersal on Mars, this data was really important to start to inform that. We did a lot of, additional surface ops, so that, again, that question of back contamination. After you've been using a tool on the Martian surface, if you want to bring it back in, is it clean enough? So on the lunar surface, we were able to develop these disinfection strategies and then use nanopore sequencing to say, okay, does it meet our thresholds? Is this clean enough to come back in? And again, I've already mentioned the biomarker tracking, the firsts of that, and the firsts in the crop production arena. So much on board the International Space Station, so much on board or on the lunar surface to get us to Mars. And I think, as you guys can tell, I could talk about any of these topics for hours, but I'm about out of time. But what I can do is you can find me later. I also have my entire team here. So I've listed kind of what their expertise is. So Sarah Rommel, Christian Mena, Hong Nguyen, Brandon Dunbar, Christian Castro, and Patrick Ridzak. And they are all here. They're around. Come ask them all the questions because this was very data light, and we definitely love to talk about our data. And just thanks to the original team of me and Sarah Rommel, Aaron Burt, and Christian John, who launched the MinION back in 2016. All the astronauts who've become amazing molecular biologists, and our colleagues at ONT, all of our supporters, Matane Jane, who's been a great collaborator over time. And just a reminder that the MinION and nanopore sequencing was critical as we went onto the moon and onto Mars. So with that, thank you.